everyone. How are you all? I'm Dr. Sumaiya. Yes, I welcome you all for today's new latest video, and today we are going to talk about bacteria. You know that these bacteria, they are unicellular organisms. They are the most ubiquitous organisms, means they are found in almost every habitat. Every habitat se we means कि like these bacteria, they can be thermophiles, they can be acidophiles, they can be halophiles. They are found in your food even, like lactobacillus, which is used for making curd. And the number of body cells are less, means that there are more bacteria present in your body than your body cells. Yes, so these bacteria, they are a type of cell which is called prokaryotic cell. What is this prokaryotic cell? This is what I am going to explain you today. So just continue with me. Yeah, this is how a bacterial cell looks like. Now, when I say a prokaryotic cell, first you need to understand the difference between the eukaryotic cell and the prokaryotic cell. My dear bachcho, you know that whenever there is a cell, there is always a membrane present which is enclosing the cell material. This is called cell membrane or plasma membrane. If there is a proper nucleus present inside, this cell is going to be a eukaryotic cell. This cell is going to be a eukaryotic cell. But if there is a cell membrane which is enclosing the material inside, and in place of a proper nucleus, there is just a DNA present, and the area where this nuclear material is present, this area is called nucleoid. So this type of cell, where in place of a proper nucleus, there is a nucleoid region where a nuclear material is present, this is called a prokaryotic cell. Now, come back to the cell. When we see a bacterial cell, like I said, the inner material is surrounded by cell membrane. Above to the cell membrane is present a cell wall. This cell wall of a bacterial cell is made up of a material which is called peptidoglycan or murine. This murine is made up of two subunits, NAM and NAG, N-acetylmuramic acid, N-acetylglucosamine. Above this cell wall is present a region which is called glycocalyx. Yes, this glycocalyx, in place of capsule, there can be a present a loose material which is called slime sheath. Capsule is present as a mode of protection to bacteria, and slime sheath is more present as a mode of adhesion of different cells. Right. Now, as a method of extension, as a method of locomotion, there are a number of extensions present. One is this. This is called a flagellum. A cell, a bacterial cell, has got a flagellum which is not of 9+2 arrangement as it is present in eukaryotic cell. Right? Apart from this, the bacterial cells, they can have number of other cytoplasmic extensions. They are called Pili or Fimbriae. They are called Pili or Fimbriae. What is this Pili or Fimbriae and what is the purpose of this? Let's understand this. let's see that two bacterial cells are present together. They have got their nuclear material, which is DNA. Abhi, I am going to tell you exact the quality of the DNA which these bacterial cells have. Okay? Now, this is the nuclear material present in both of these cells. Now, if I say that the nuclear material of this wala cell, the nuclear material of this wala cell is going to pass on this side, right? So, it is either going to get a part of this cell or either going to present as some extra nuclear material, right? Suppose this is A, this is B. So, from this A, the nuclear material is being passed to the B side, right? So, this A wala ko we will be calling donor cell, right? And this B wala, which is receiving the nuclear material, we will be calling it acceptor cell or recipient cell. Tick. Also, you can call this male and this wala female. Tick. The tube here which you are watching, this is a cylindrical tube. Actually, this is made up of a protein, pilin. And yes, this is a bridge which is exactly formed by the pili, right? So, this bridge ko now a proper name we are going to give. This is called conjugation bridge. This is called conjugation bridge, right? This is called conjugation bridge. Now, through this conjugation bridge, a nuclear material of a donor cell is being passed to the acceptor cell and the overall process is called conjugation. This process of conjugation was discovered by Lederberg and Tatum. Now, this is also a method of horizontal gene transfer, HGT. Now, we have seen two types of extensions. One is the pili or the fimri, and the true pili in fact is used for conjugation, which is seen more in the gram-negative cells. Abhi further I am going to explain you about gram-negative cells also. So, just have patience and continue with me. Now, this is the flagella, right? This flagella, the protein with which it is made up of is flagellin, and the protein with which the pili is made up of is pilin. Okay? Now, these pili are not a mode of locomotion. They are used as a thing for cell-to-cell attachment for the purpose of colony formation or as I have shown you for conjugation. Okay? Now, these prokaryotic cells, they don't have any of a eukaryotic organelle except ribosomes. That too is having a difference. So, let us see a prokaryotic ribosome and compare it with a eukaryotic ribosome. A prokaryotic ribosome, as you see, a complete ribosome is shown here, is a 70S ribosome. S7 means Swedberg unit. This 70S is divided into two subunits, the larger subunit and the smaller subunit. This larger subunit here is of 50S and smaller subunit is of 30S, right? But if you see a eukaryotic cell, here the two subunits are of 60S and 40S, and overall it is a 80S ribosome in a eukaryote. I hope that this point is clear. Okay. Now, these bacterial cells, right? These bacterial cells, this is the cell membrane. Their cell membrane can be protruded inside and like it is trying to give out some more extra space to the cell, and these inward extensions, they are called mesosomes. Mesosomes. What is mesosome? As you know, okay, they are the bacterial cells, they are the prokaryotic cells, right? So they don't have any of the eukaryotic organelle, right? And in eukaryotic cells, for the purpose of respiration, energy production, there is mitochondria present. So here in place of that, the inward projection of plasma membrane is present, which is called mesosome. So these mesosome not just participate in cell respiration, but also they are somehow linked with the cell division. Let me tell you that these bacterial cells, they divide through a type of method which is called binary fission. Binary fission is the most famous method for their cell division, right? So whenever, suppose this is one cell, right? This is the nuclear material of this cell, and see, this is the mesosome present. So this mesosome is actually helping in the process of binary fission, right? Now when the nuclear material will divide, and the cell will also divide into its respective daughter cells, right? Now the question here arises, okay, what is the type of material, nuclear material, which is present in a bacterial cell and how it is different from eukaryotic cell? Beta, these bacterial cells, as I have told you, they don't have any nucleus. And in the eukaryotic cell, like suppose if this is a eukaryotic cell, so this eukaryotic cell is going to have the nuclear material inside, like there are chromosomes present. Sometime you could have seen nucleolus, right, which is the site of ribosome assembly, and number of ribonucleic acids are present there. But here in the prokaryotic cell, this type of thing is not present, right? So in a prokaryotic cell, this DNA is circular in shape, right? And that too is just a single piece and is double-stranded DNA. Suppose this is one strand of DNA and this is the second strand of DNA. So this is, I am showing a linear DNA, which is a characteristic of eukaryotes. But in prokaryotic cell, a bacterial cell, a circular DNA is present. So we will be marking it as a single circular double-stranded DNA. So in most of the bacterial cell, this type of DNA is present, and again I am telling you this thing, okay, this area where this DNA is present, this area is termed nucleoid. This area is termed nucleoid. So in place of nucleus, यहाँ पे you are getting a nucleoid region, right? So there is one more important thing to consider here, like when the nucleus was present, which was having some nuclear pores in the eukaryotic cell, like the DNA replication was taking place inside, right? Then transcription was also taking place inside, and from these nuclear pores, the mRNA transcript was coming out, right? Here in these prokaryotic cells, there is no such nuclear membrane present, right? No nuclear pore present. So whatever is taking place in the cytoplasm only, right? So suppose this is the mRNA transcript present, right? This is the mRNA transcript present, and on these mRNA transcripts, a number of ribosomes, they will come together and they will join. First smaller subunit and then larger subunit, and this will give a type of structure which is called polyribosome. This is a question of NEET also. Okay, this polyribosome, in fact in NEET it is called as polysome. They have asked you as polysome. So yes, this is that polysome. Polysome, right? So in the prokaryotic cells, number of ribosomes, they come together, they join on one mRNA transcript, and then the process of this protein formation continues from this. Now, come back to the main cell. So, you know, okay, as an extra chromosomal material, right, some plasmids can be present. These plasmids, they provide a type of resistance, a type of antibiotic resistance or some special factor to the cell, right? So we have covered a lot of material about these prokaryotic cells. Now I hope now you are aware about the cell membrane, about the cell wall, about the glycocalyx, the flagella, the pili, the fimbriae, the plasmid and the ribosomes, and moreover you also know that nuclear material here is a single circular double-stranded DNA, right, which is present in the region which is termed nucleoid. Now, if you are asked how many chromosomes a bacterial cell have, then whenever you know a chromosome assembly takes place, so from the DNA this assembly takes place. So in the eukaryotic cell, that chromosome binds with some histone proteins. Here the DNA will make a binding with some non-histone proteins. And since here it is just a single piece of DNA, so there is going to be a single chromosome here. So this means that bacterial cells, they are haploid in nature. One more important thing I have to discuss with you is the classification of the bacterial cells as per their shapes. My dear bachcho, there can be a number of shapes present for bacteria, right? So if they are spherical, then to this you are calling a coccus, right? If they are spherical, to this you will call a coccus. If two of the coccus they unite, it is called diplococci. And yes, there are number of bacteria like Diplococcus pneumoniae, right, which is a causal organism for pneumonia. Now, if they form this chain type of structure, right, number of coccus, they are joining to form a chain type of structure. So this is called streptococci, right? Streptococcus pneumonia, Streptococcus pyrogens, number of bacteria are like having these chains. Now, if they are in a unit of these 4, this is called tetrad, and if a unit of 8, then it is called sarcina, right? And if they form a group which is somehow a cluster, which is somehow looking like a grape, then it is called staphylococci. One example is Staphylococcus aureus. Now, if these bacterial cells, they are having some rod shape, okay? So it is called rod shaped bacteria commonly, or to that you also call bacillus, right? Now, if 2 of the bacillus, then just like cocci, you can call it diplobacilli. If a chain of bacilli, just like streptococci, you will be calling it streptobacilli, right? These bacillus, they can have flagellas, right? It is a characteristic of some of the bacillus cells to form spores inside, like Clostridium botulinum, Clostridium tetani. Clostridium botulinum is responsible for botulism, a food poisoning disorder. Clostridium tetani is responsible for tetanus, right? So there are some spore forming bacteria, and their spores, you know, they are so resistant that they are not killed by simple boiling. For this they need autoclaving or pasteurization, right? So what happened in this spore formation, their protoplasmic material is like constricted over this area only, and this is a resistant spore, and inside the nuclear material is present, and then after some time the outer material, outer wall get degenerate. So this is how a spore is left out in unfavorable conditions, and when they get the favorable chance, then though they are not going to leave anything. Apart from this very famous coccus and bacillus, there can be some other shapes also, right? Like If it is comma shape, so it is Vibrio, right? It is called Vibrio. Vibrio say, you must be remembering Vibrio cholerae, the causal organism of cholera, right? There can be some stiff type of spirals, right? To which you will be calling spirillum. If plural, then spirilla. So, Heliobacter pylori. If they are somehow not so rigid, right, little bit elastic in nature, so then they can form more spiralated character, right. They can also have tuft of flagellas at the end. So this is called spirochetes, and one spirochete example is a very famous as a STD causing bacteria, which is Treponema pallidum. Treponema pallidum, you know, it causes syphilis. So these are some of the classification of bacteria as per their shape. Now, number of time in front of you I took the name gram positive or gram negative. So how you can decide gram positive or a gram negative cell? This all you are deciding on the basis of their cell wall. So there was a scientist, the name of that scientist is Christian Gram, and that gentleman, what he do, he actually stained bacteria in different stains. So first he stained bacteria in crystal violet stain, right? So those all of the bacteria, they take on that crystal violet stain. Take after that he washed them with alcohol, and many of the bacteria, they lose that stain. So then as a counter stain, he used safranine, which gives out a pink color, or you can also use malachite, which gives out a green color. So some of the bacteria, they took that pink stain, counter stain, right? So those bacteria, those who have got, those who retain this purple violet color, they are called gram positive, and those who lose that purple violet color and in return they take the counter stain, they are called gram negative bacteria. Right? Now, as I am saying, there is a difference which is in their cell wall. Okay? The difference is in their cell wall. You know, the cell wall of the bacteria is made up of peptidoglycan, right? Just have a look. Two cells are in front of you. This side is gram positive and this side is gram negative. You see, on the side of this gram positive, the peptidoglycan cell wall is a thick cell wall, or in the side of this gram negative, the peptidoglycan cell wall is a thin cell wall, right? Peptidoglycans, they are made up of NAM and NAG associated with some proteins. So here in this gram positive side, this is more present, like 70 to 80% of the peptidoglycan or murine is present on gram positive side, and just 40 to 50% of the murine is present on gram negative side. Other than that, these gram negative bacteria, they have got an outer layer, an outer membrane. These can be of some lipoproteins and lipopolysaccharides. So lipo word is involved here. Lipo means for lipid. This is the lipid which is exactly providing them resistance. And you know, these gram negative wala are more virulent. They are more virulent. They are more pathogenic, right? So these gram positive wala cells, they can also have some teichoic acids or lipoteichoic acids present with them along with some surface proteins. Here also surface proteins are present. Apart from this, some porins are also present, which are providing the inward nutrient intake by the gram negative cells. These gram negative, as I said, are more pathogenic, right? But for your knowledge, let me tell you, you know Mycobacterium tuberculei? That is a gram positive bacteria, but it is the world famous pathogenic bacteria. So what it is having a waxy coating of mycolic acid on the gram positive structure, which is providing it a defense, right? So these were some interesting facts regarding the bacterial cells. I hope you enjoyed these facts and the topic is clear to you, right? So we will be meeting in our next video, and let's see in the next video. Take care everyone.